All right, there we go. And now for something completely different. So you may be wondering why an endocrinologist who studies mostly beta cell biology will be talking to you today about metabolomics, and hopefully I'll be able to explain why that is. You may walk out if you're wondering the same question. But what I hope to do is tell you about some of the work in metabolic pathways that we've been discovering in the pancreatic beta cell involved in glucose sensing, how that has led us to develop specific tools for assessing metabolic flux in cells, and then at the very end, one brief case of how we're applying it in a cancer system. And so with that, I have no disclosures related to this with the exception of some grant support from the Cancer Center, so anything I say will be overinflated in terms of its importance. And what I'd like to tell you about is some of the similarities between beta cells and cancers. They're both metabolic tissues, right? Beta cells have high rates of glycolysis and not so well known high rates of glutaminolysis, very similar to cancer cells. They are one of the differentiated tissues that have very high expression levels of PKM2, sometimes has been known as the cancer isoform of pyruvate kinase, but it's the normal pyruvate kinase present in these cells. They also have a lot of cytosolic isocitrate dehydrogenase metabolism, again a key feature of some cancers. And both of these tissues in many circumstances are thought of as being very ATP dependent. Their metabolism is developed so that they can rapidly make and use ATP. But there are two big differences about these cells or tissue types, and one is that beta cells are one of the have one of the lowest rates of turnover to the point that once you're an adult there is zero turnover of these cells, yet they have all this same equipment that's commonly found in very metabolically active cells. And I don't think I need to tell this group this much about Warburg metabolism and that type of stuff, but it's worth a reminder that differentiated tissues are believed to generate most of their ATP from oxidative phosphorylation through the mitochondria, and very little generally through more anaerobic glycolytic metabolism. And the main reason is it's very efficient. You get about 36 moles of ATP per mole of glucose if you're completely coupled. If you're doing purely anaerobic glycolysis, it's very low yields. It's 2 moles of ATP per mole of glucose. So if that's the case, why do cancers have so much of this? And I still think that is an unanswered question. One of the thoughts is that, well, there's not much oxygen, so you can make a lot of ATP that way, but there may be much more to that. And I'd also like to point out that while these arrows are big in cancer metabolism and that this is only 5%, remember these yields. You're still getting a heck of a lot of ATP from oxidative phosphorylation that you're not getting you're not getting from the Warburg type of metabolism. And again, back to the beta cells, both of them have these interesting PKM2 isoforms, which potentially highlights the importance of phospholynyl pyruvate in this metabolism. So the canonical view of how beta cells sense glucose is through metabolism. You metabolize, you make ATP. The ATP closes the KTP channel. That leads to an influx of calcium into the cell, and oops, wrong direction. And that causes the insulin granules to fuse with the cell membrane and insulin release. But is that really the whole story? And a lot of questions started happening when you knocked out the KTP channel. And when you did that, you can still get very robust glucose-induced insulin secretion. Again, this is completely lacking the ATP sensing mechanism. And not only that, you can also get the same glucose-induced calcium oscillations present, suggesting that maybe ATP is not the whole story. So a number of investigators started proposing additional hypotheses of metabolic pathways that might really be important for sensing glucose. And I'm not going to go through the details of them, except to say that there are a lot of them. They're ultimately involved in synthetic pathways that are needed to generate things like NADPH, which are essential for cell growth and division, and yet these are very mitotically inactive cell type. But one of the real common facets of this is this arrow right there, which is the anaplerosis arrow, which just means you're adding carbon to the mitochondria in a non-oxidative way, and that allows you to do synthesis. So if you have a cell that's supposed to only make ATP, why do you have this synthetic pathway that's actually depleting ATP relative to the amount of glucose that's going through? And it suggests that there was something else going on. And so our hypothesis in the beta cell is that it's not all about ATP. There are other sensing mechanisms. And we started to try to understand this from a very rare mutation that's found in children who develop hypoglycemia at around age 6 to 1 year of life following a protein-rich meal. Glucose doesn't make them hypoglycemic, fat doesn't, but protein does. And interestingly, another aspect of this disease is that they have ammonia levels in the 100 to 200 range and completely asymptomatic from it. Well, the mutation was identified in glutamate dehydrogenase. And the kids who have this mutation are no longer able to have this enzyme inhibited by mitochondrial GTP. And that prevents glutamate from entering the TCA cycle. And so, in essence, what happens, they eat protein, and all the protein carbons flood into the TCA cycle and presumably stimulate insulin secretion. I keep going backwards, so sorry about that. So we thought that GTP might have an important sensing role, and to understand GTP in the mitochondria, you have to put it in contrast to ATP in the mitochondria, which we know pretty well. It's mostly made by oxidative phosphorylation and from the oxidation of things like NADH and FADH2 in the mitochondria, and that ATP is rapidly transported out of the mitochondria. And the stoichiometry of this is very loose, and that's because this is dependent on the coupling of protons translocated through here. If you're uncoupled, you make less ATP. So maybe not a great energy sensor. GTP is very different. It's made directly by the TCA cycle in proportion to the rate that the TCA cycle is turning, and it's also a trapped metabolic pool. It doesn't exchange with the cytosol. And so the whole idea is that this GTP production is a way for the mitochondria to know what metabolism is going on. And it turns out that the enzyme that does it, succinyl-CoA synthetase, has two isoforms, one that's GDP specific and one that's ADP specific. And cells that are more synthetic have the GTP isoform. Cells that are more oxidative have the ATP isoform. And it's the ratio of these two isoforms that determines the amount of GTP that's being made in the mitochondria. So I'm not going to go through all the work that led to us figuring out this whole metabolic pathway, except to say that we think it ties together that yellow arrow I showed you before of anaplerosis with the TCA cycle, where you get a stoichiometric production of GTP, and that's combining with oxaloacetate made by this enzyme in beta cells to make phospholinopyruvate. And that phospholinopyruvate is coming out of the mitochondria because it does have channels that can escape. And this is the highest energy phosphate in the cell. It's much higher than ATP, and it's metabolized by this cancer isoform PKM2. So that was all the precursor to why we started developing these techniques, because we're proposing something that's quite different than the normal expectation of how metabolism is working, and we had to develop the tools to be able to sort this out. So one of the tools we were able to do is use mass spectrometry to be able to measure these fluxes. And the basic idea is that you can put carbon-13 into a substrate in a very specific position, and that carbon-13 weighs 1 more Dalton than carbon-12. And so by mass spec, if you can separate things out, you can measure the relative rates of carbon flux through a particular pathway. And so in this case, we used 113C labeled pyruvate which goes in the TCA cycle, and whether it passes through TCA cycle or pyruvate carboxylase, it gives this particular labeling pattern. And then simply by knowing the relative enrichment in this pathway versus the precursor versus the product pool, we can directly calculate the rate of flux through these pathways at metabolic steady state. And what we found is that this rate of flux is very, very high in beta cells. Up to 1/3 of glycolysis is going through this pathway that is not going into ATP production, and that flux is increasing with increasing glucose concentrations. If you knock out any of the parts of this pathway, you completely lose insulin secretion, no matter how high the glucose concentration is. So it's a very active metabolic flux that's important for insulin secreting cells. And that's good, and that would be great if there weren't other metabolic fluxes, but there are. And there are a lot of things coming in and out of the mitochondria that we really want to know what's going on. And a lot of these fluxes intersect. And you can't just look at a single metabolite and know what's going on. So, and for instance, if we take our cells and we incubate them at different glucose concentrations, we get different insulin secretion rates. And if we were to look at the metabolites in the different pathways at these different glucose concentrations, we'd see that most of them go up, but we can't distinguish what this is telling us about which metabolic pathways are active. So that's where we developed a much more involved labeling schema. And this is based on work done initially in MR, where you're using uniformly labeled glucose with all carbons in it 13C labeled. And for instance, if the 13C is in the blue pyruvate up there, as it goes into the TCA cycle, it's going to label specifically these carbons of alpha-ketoglutarate or glutamate. Or if it goes through this pyruvate carboxylase, it's going to give you these, and then those will join into to give a double labeling pattern. And if you turn the cycle a couple more times, you start to generate all these different patterns. But it actually turns out that it's possible to deconvolute these relatively straightforwardly by with some degree of specificity by NMR by just looking at the glutamate. What we thought would be cool is if we could not just look at glutamate, but look at every single metabolite in the metabolic pathway, track every single carbon in all the positions, and know exactly which fluxes are active. And that's precisely what you need to be able to unravel all these different metabolic fluxes. And the basic labeling pattern is you have a metabolic steady state that you use to with unlabeled glucose, and then you change from unlabeled to labeled glucose, and you just collect different time points and you put it in the mass spectrometer. And so we've been very successful now looking at flux through these pathways. So for instance, just trucking through glycolysis, we can follow glucose. This is time, and this is the amount of enrichment of our label across time. Glucose going into fructose 1,6-bisphosphate. We can track it going into the glycerol 3-phosphate shuttle, and we can actually get the relative shuttle rates relative to glycolysis from this. We can follow it down to 3-PG, 2-PG, on into phosphoenol pyruvate, down into pyruvate. We can get into lactate. We can follow it into the pentose pathway. Not only can we get pentose flux, we can tell if it's going into a ribose synthesizing pathway, an NADPH synthesizing pathway, or both, which are really crucial to understanding metabolic growth and turnover. And we can also look to see if it's growing in serine, and we found that very, very, very little was going into serine at all. So it gives us a lot of information of what metabolic flux is doing, and here I'm just zooming in on the early time points of these curves to show that we can look stepwise at metabolism. And what's really interesting in these beta cells is that it's not all reaching 100%. You would think if all you're doing is metabolizing glucose, everything would go to 100%, but they're dilutions, and these dilutions are important. They tell you that carbons are entering a different pathway. We can go from glucose into pyruvate and then follow these things into the mitochondria. And we can follow the label tracking into acetyl-CoA and into these position-specific portions of citrate and glutamate, which is shown in this curve. We can watch it go through this synthetic pathway, which is this curve. we can watch these carbons come back around going through the TCA cycle, or even second cycle TCA flux going through here. And the importance is not really knowing what all these pathways are doing, but that we're getting very, very detailed precision of metabolic fluxes in these cells. And this is just stacking everything on top of each other, going from pyruvate, or phosphenolpyruvate to pyruvate, acetyl-CoA, citrate, and then, very interesting, glutamate. And so each of these rates of buildup tells you the flux, and each of these tells you the relative source of the carbons going into that pathway. And that brings us to why we want to do flux and just not measure metabolites. And that's because metabolite concentration really misleads you as to what's going on. So if we look at the rate of enrichment of phosphenolpyruvate at low glucose, it's this. And as we increase the glucose concentration, those enrichments go up. But if you look at the actual concentration of the metabolites, where we've gone from a doubling of the rate of enrichment, we're seeing no change in the concentration of phosphenolpyruvate. And here, we're seeing no difference in the rate of labeling, but we're seeing a doubling in the concentration of the metabolite. And it really takes a combination of putting the label incorporation rate plus the concentration to be able to get the absolute fluxes. And we're able to calculate these through a collaboration with Graham Mason in the radiology, who's developed this program called C-Wave that allows us to put all this mass spec data into it and get these relative fluxes. And what I'm showing you are in the blue lines are the modeled data, and the red squares are the actual raw data, and we're getting very nice fits for all these fluxes. And so we believe that we're able to, with a very high degree of specificity, determine the absolute rate of fluxes through these pathways, which then gives us a very different picture of what metabolism is. And I'm not, again, go through the details of the different pathways that we're looking at. Just want to emphasize that we are able now to measure these different metabolic rates through pathways that seemingly overlap and intersect. Which takes us to the final cancer slide, where we're doing this in melanoma cells, and this is work that we've been doing with Marcus Bosenberg and a graduate student in his lab, Nick Theodosakis, and we're looking at melanoma cells, and we're treating them with Vemurafenib. And I'm not going to pretend to understand melanoma biochemistry or how these BRAF inhibitors are actually working, but to give you an example of how this data can shed some light on what's going on. So these inhibitors really slow down metabolism in these cells and inhibit cell growth. And so if that's the case, where is the block in metabolism? And when we start looking at that, we see some interesting results that don't really match up with some of the initial expectations, at least my understanding of what the initial expectations are. So 3-phosphoglycerate is one of the trioses in glycolysis. It's relatively high up. And what you actually see is an increase in the concentration in that metabolite, a very large increase. And if you look at the rate of flux incorporation into it, it's not distinguishable from the control cell, suggesting that that the early glycolytic flux may actually be faster in these cells treated with this particular inhibitor, or we just may not have resolved this very steep initial curve. When we go a little further down into glycolysis, looking at phosphenolpyruvate, again, we see that the concentration of this metabolite is increased, which doesn't go with at least a very early block in metabolism. And while this rate of incorporation is apparently slower, if you correct for the fact that this pool is bigger, you're actually seeing the same metabolic rate. When we get down to lactate, the concentrations are no different. And the labeling pattern does seem to be slower. Again, if you account for the fact that this pool is less labeled, this still does seem to underperform, and it does seem that metabolism into lactate is slower, but that wasn't where a lot of the dramatic results were. These were once you got into the TCA cycle. First of all, note that the blue curve is the amount of glucose carbons going into the TCA cycle. And you can see that there's some other carbon source that's predominantly going into the TCA cycle besides glucose that's driving its flux. And second, that there's absolutely no detectable label going into the TCA cycle at this level. And there is a modest reduction in glutamate concentrations, but if you go even further around the TCA cycle, you see a complete loss of carbon flux into malate. And what this suggests is that maybe the major effect of this is, sure, it is potentially slowing Warburg metabolism down, but it really seems like it's stealing all the carbon from the mitochondria and taking it to another pathway. And that may be an unexpected mechanism of how these types of drugs are working. And so we're hoping to be able to apply this not only to this cell model, but other cell models of trying to understand either disease or drug action or general metabolism to get a good sense of what's going on. And we're extending it beyond glycolysis and the TCA into a lot of these other pathways that we all had once upon a time in college biochemistry and have forgotten about since. So in conclusion, I didn't show you much data about this, but I'd just like to say that I think the mitochondrial GTP cycle is an important mitochondrial metabolic sensor. In cells where we've accelerated this pathway, we see accelerated rates of cell growth, and the opposite happens if it's decelerated. That mass spectroscopy-based carbon tracking is an important new tool to assess very discrete metabolic fluxes in cells to a degree of resolution that's not been achievable before. And finally, that ATP synthesis may not be the pinch point controlling insulin release or cell growth. That a lot of the generalizations that we have about how metabolism in different cells may not be quite true. A lot of the work was based in muscle, and muscle is not all other tissues. And so with that, I thank you for your attention, and I'd like to thank especially Tiago, who did a lot of the labeling studies, Graham, who helped with the modeling, Marcus and Nick for the cancer work, and Matt has been an assist doing some work that we didn't talk about today. Thank you.
So I think we're all channeling our inner Leninger after hearing that presentation. You have to talk rather than into it. And eventually it comes on. Maybe.
Can you mention the Cancer Metabolomics Working Group?
Indeed. There is a Cancer Metabolomics Working Group that Marcus, me, and Xiaoyang Yang have been putting together, and it meets once a month on Wednesday afternoons, and it's a combination of research in progress as well as journal clubs, and everyone's invited. There's food. And it's kindly supported by the Cancer Center. It's really actually been quite a big success. It's very well attended and a lot of very interesting work presented there.
Last question for Dr. Sklar. If you speak really loudly, does it work?
So I wonder if you've had an opportunity to test a range of cancer cells with these methodologies.
We have not as of yet, and that's work that we hope to be able to get funding for at some point to be able to do. And what I actually would really be interested in doing is comparing not just different cancers, but comparing normal tissue to a cancer tissue, because I think there's a very big gap, and that is something that we're doing, because one of our strong working models is insulinoma cells, and we are working with human pancreatic islets doing this metabolism.
Let me ask the question slightly. So have you looked at multiple examples of melanoma?
No.
Or is this all based on this?
This is very early work that we've been doing in collaboration with Marcus.
So my real goal is to determine whether you feel that you have a signature that could discriminate reliably cancer cells from normal cells, or transform cells from immortalized cells that are not fully transformed?
I would say absolutely not at this point in time. And we don't have the normal tissue to really be able to compare, but I think that would be a goal, and like to say that we can get some of these measurements from as few as 5,000 cells, so it might be possible to do these things from human biopsies.
Dr. Kibbey, thank you very much. Fabulous presentation.